done a series of small projects. The first problem that was encountered when I started the project was when we added the phosphate buffer solution to blood cells, we ended up with the weird star shape. You can see how that looks compared to normal cells. It's obviously not right. The two most obvious causes that I thought would be an unknown disease in the donor blood, but I outruled that problem by using different blood samples from healthy donors. So it was concluded that it must be a problem with the buffer. I used a few different buffers and I made tables comparing different components of the buffers, and this is just one example of one. The first buffer that I used, I looked at the date and whether or not it contained calcium chloride and magnesium chloride, and you can see the cell irregularity that happened with the first two. And then I said variable with the third one because there were certain dilutions of that one where the cells looked normal. Here is the second buffer that I used, and the problem still occurred. This is the new buffer, the last buffer that we used, and different dilutions. And it goes from left to right, and then across the rows, and then down. You can see kind of a strange pattern. We had the crenation in the 0.5x, the 2x, the 3x, but then not in the other ones. And we figured out it had something to do with the osmotic pressure. This is a hemocytometer, and that's what I used to count cells. The picture on the top right is what the hemocytometer looks like, and then on the bottom left picture you can see how the outside four corners have a bigger grid in them. The target amount of total blood cells per microliter that I was trying to get to was 1,000, and I tried a bunch of different samples and found that 0.5 microliters of blood for every 1,000 microliters of PBS, and using that with a 1-to-1 ratio of blue dye, I would get that amount. Another thing that I did was counting CHA cells, which are the cells from a cervical cancer patient. Jenny had some, and I was lucky enough to get a look at those. And the third project is fluorescent labeling of white blood cell nuclei. And for this, I used a HOCHUS dye, which stains the nucleus of white blood cells. And I used concentrations between 0.2 and 5 micrograms per milliliter, and incubated them for 20 to 30 minutes. I did 3 trials. The first one was 0.2 micrograms per milliliter. I did 1 microgram per milliliter and 5. The third part is the centrifugation, and the centrifugation theory is that materials suspended in liquid matter will separate because of the effect that gravity has on them. In conclusion, we still don't know the cause of the star-shaped cells, but we found an acceptable buffer solution that works. And I was informed by Jen the other day that all of these experiments relate to the project Sample Preparation for Gene Expression-Based Biodosimetry. And this means finding the expression profile of white blood cells without red blood cell RNA contamination.